And today I'm going to talk about 5 of things in cancer, which mean what can we do using phylogenetic concepts and methods and tools to understand cancer evolution, because cancer, it is evolution. So I'm going to give kind of like a soft, shallow talk overview, because my feeling is that most of you are not familiar with the concepts of cancer evolution. So bear with me for a while. I'll give you some introduction to basic concepts. I will be fast, but interrupt me at any time. I'll try to explain things that what it matters that you understand what I'm trying to say. But I guess that we all know what is cancer, which is uncontrolled cell proliferation. This results in tumors, and when these tumors invade surrounding tissues, they become malignant, and then sometimes they're able to go into the circulation, lymphatic or blood, and colonize other niches. It's called metastasis. In principle, let's think this way. Cancer is a genetic, if you want, epigenetic disease, and is the result of mutations, which we call them driver mutations in genes, which are responsible for things like mainly cell cycle control or DNA repair, and they affect the function of the cell, and that cell starts proliferating in an uncontrolled manner. Okay? So we are talking about driver mutations in driver genes. The errors that cause these mutations are most of the time sporadic. They are due to just biochemical dominations, UV light, tobacco, DNA replication. Most cancers are sporadic. We distinguish here about somatic mutations. In the next slide, I'll show you that we have all the time mutations, a lot of them in our body. Most of them, they do nothing, right? But they are not in all the cells, right? And sometimes they hit one of these driver genes, and we get a cancer. It's something like that. From the very first moment we're a zygote, we start accumulating mutations, right? Through the development of the embryo as a baby, we accumulate mutations. Sometimes some of them, they hit some genes that provoke some uncontrolled growth, but that's not still a cancer. There are expansions of clones, identical cells genetically, and they don't do a cancer. But sometimes we hit other genes, and then we get a tumor, and then we get other things, right, going on. So there is a continuous process of mutations, and trust me on this number. Every second we accumulate, all of you in this room, since I start talking, you have accumulated 10,000 million mutations in your body. Don't worry. By far, most of them are not deleterious, right? But that's the truth. We get a lot of mutations. A different thing is whether we are able to detect them, right? But we have better and better techniques to detect mutations, even if they are very infrequent. Just doing bulk sequencing, the standard sequencing, we can now detect mutations under a 100% frequency, or even 1 on 1,000 frequency, right? And even more than that. We have high fidelity, amplification techniques and sequencing techniques, and so on. So we can detect a lot of mutations. In tumors, this results in what for evolutionary biologists is quite obvious, in heterogeneity, right? And we have what we can call clones. I have next slide explain a little bit what is a clone, because it's a term which is not always very clear, but basically is a set of populations. We have the same mutations at the driver genes. Let's look at this clone. So we have a red clone, a blue clone, even with a single tumor. Every tumor is different in every person. Tumors are, the name of a cancer is just the tissue of origin, it's colon cancers, but every colon cancer is different, right? Sometimes they share some mutations, sometimes they don't. But at the end, we have a single tumor is a population, right? With heterogeneity. This is called intratumor heterogeneity in the community. What is a clone? Well, a clone for us, for evolutionary biology, is very obvious what it is, because we think this way all the time. That's not the truth in the cancer community. But let's take so far for this talk that a clone is a population of cells that share a common ancestor, which we call a clade, a monophyletic group, right? And they share some of these important mutations, the driver mutations. When they accumulate they are called passenger mutations in cancer, right? Because there is no recombination, so there is the driver and the passenger of this linked genome, of this big bus. Well, sometimes this is not clear in the literature, but let's think this way. Here you get a driver mutation, which is a red one, a blue, and of course you have sampling and so on, but we'll go to that later. Okay? But the context is this: a clone, set of driver mutations. Indeed, cancer is evolution. So we start from a healthy cell that accumulates one of these driver mutations. Then this clone accumulates other passenger mutations, so we keep the same color. Then we have some selective pressure, which might be the immune system at the very beginning. Some clone that acquire a novel mutation here is able to survive, then keeps evolving, novel driver mutations here, and expansion. And then there is migration, okay? Which is called metastasis in cancer. Right? To migrations to a different organ, a different ecosystem, and we can have, for example, another selective pressure, in this case therapy, and there is a minor clone here that is the only one that is able to resist this therapy, and then we have like a recurrence. Okay? So cancer is just evolution. This could be finches, if you want, or any population in the world, but it's just cells. So cancer is exactly an evolutionary process in which selection is something deemed as extremely important. We can see it this way. We can see that we have germline evolution, species evolution. As far as I know, is all of you work at that level, right? What we call evolution, simple evolution. But trust me, we can see that there is also somatic evolution. So within a human population, one individual has trillions of cells, and some cells sometimes, in principle, just one, right, is the common ancestor of a tumor mass, right? So we have a continuous of evolution, and there are indeed some differences, mainly related to the fact that somatic evolution is asexual, okay? And the time scale is different. It's just decades, if you want. There are several challenges to study cancer evolution. That one of them is this short evolutionary time. You might think that we have lack of genetic variation, but I just told you that we accumulate 500 million mutations per second in our body. Okay? Not in the same cell, indeed. Right? And as far as we get better techniques, and better techniques, we are able to distinguish more and more of these mutations. So probably this is not a challenge any longer. No recombination. No recombination is good or bad. Okay? I mean, we know that if we have recombination, we have multiple instances, multiple replicates of the evolutionary processes at different non-recombining blocks, right? So we get better estimates of the variance of this process. When we have just a single locus, then variance becomes very important. And that's the whole genome is a single locus in cancer evolution. Okay? Positive selection is assumed to be extremely important. If you think of the DN-DS ratio between human and chimps, this is 0.2. In cancer, it's 1 across the genome. There is no, with some exceptions, negative selection in cancer. Any cancer cell can become a monster, have 20 copies of this chromosome and miss 7 chromosomes, and still survives. Why? Because it only has one mission, one function, to divide. Doesn't need to do anything else, right? So many genes become accessory. Okay? There is redundancy of functional redundancy in the genome, right? But this is the truth. Okay? There might be some instances of some deleterious mutation. We hope that, if we have deleterious mutation, we have some target, right? But no, they can do anything. They can accumulate any kind of structural rearrangement that nothing happens to that cell, keeps dividing. Okay? There is no meiosis. You don't need to have some order segregation. So it can do whatever a cancer cell. And another difference is that we have a lot of structural variation, much more than in species evolution. This is what I just said. You get all kinds of rearrangements. You get chromotripsis. The chromosomes get broken in 1,000 parts. Then they join. You have confetti chromosomes all over the place. The cell still divides. So we have this hope for monsters, even much more common than in species evolution. And I think this is totally true. And as you know, we have poor evolutionary models for structural variance. Well, if you disagree, we can talk about this later. I'm going to put some slides about this. We don't use structural variation to infer evolution, really. Just one paper, 2 papers, 3 papers. We don't do. We keep using single mutations. Right? Why? Well, I'm going to give you some reasons. And then something which is also very important is that when we sequence a tumor, we're sequencing the whole population of cells. we are sequencing millions of cells. That means millions of individuals. The equivalent in species evolution is what we call pool sampling, if you know what it is, pool sequencing, right? When you get 100 Drosophila individuals from the same location, and then you put them in the same tube, and you extract the DNA from the whole set, it's just for money, okay? To afford money. There is like a prior belief that this population is a clade, so you have different clades in different locations, so you can pull the individuals from the same location, thinking that they are independent evolutionary units. That might be okay. The only reason is time and money to do this. In cancer, we cannot escape, or we couldn't until very recently. So this is how we sample. I know that there are many species which are populations very difficult to sample, but you can imagine that cancer is a big problem for several reasons, because there are ethical issues, there are technical issues to sample a tumor, and you need to rely on third party, on pathologists, surgeons. So the logistics are very complex, right? We get what we get, pretty much. But basically, we have this bulk sequencing, which is the standard one. You get a piece, a biopsy, you sequence that, and you're sequencing millions of individuals that belong to different clones, right? So this is like a pool thing, right? A mixed thing. Sometimes you can get multi-regional samples. In some type of cancers, it's easier, right? Depends on the type of cancer. Of course, you can be blood, right? Also, leukemias, and sometimes you are able to sample, particularly in more advanced cancer, circulating tumor cells and circulated tumor DNA from body fluids, mainly blood, but also saliva, urine, other type of blood. The concept of purity is very important here because we are sampled these, but we are getting healthy cells also. Okay, so we get a mixture. So our estimates of allele frequencies need to be corrected for this purity. Purity is the amount of cancer cells that you have in your sample. That's how we call it. We always try to get also a healthy sample, which often is saliva or blood, lymphocytes, because we need to distinguish germline from somatic mutation. So we have always this match from the same individual, like normal tissue and cancer tissue. The type of mutations that we have in cancer is a lot of different ones. Rearrangement gains and losses are extremely common in many cancers. All kind of stuff can happen, but I will argue that single nucleotide variants are the best ones we can use so far to estimate evolutionary parameters, because we have very clear models, right, for single nucleotides from A to T, C to G. They are very old. We know how they work. There is a lot of models to accomplish many things with these models. Copy numbers are very important in most cancers, extremely, right? There are a lot of copy numbers, but the problem here is that they overlap. Therefore, they are not IID any longer. They are not identical independent distribute these things. And you get the same pattern, right, of copy numbers. If you segment these, normally what do you do segmentation, you identify breakpoints, and then you get these kind of numbers, 2 copies, 2 copies, 3 copies, blah, blah, blah. But the same string can be explained by different patterns. So when you have overlapping events, things becomes a nightmare, right? We heard about the TFK model yesterday. That's from 91, and we are not using that, sorry, so in phylogenetics, really. At least not commonly, right? Why? Because of this. Because there is a problem of identifiability. There are all infinite explanations from the same pattern. So I will focus on this. In our group, we have been focusing on single nucleotide variants just because of these reasons. But it's, it's a pity because it's very abundant and it will be very informative, copy numbers, right? But homoplasy here is very common for these type of events because of this overlapping nature. In the cancer community, most of the models assume this, assumes the infinite site models that mutations is rare, that are not reversible. They only happen once, right? Because the underlying thought is that mutation is rare, so therefore it never happened twice. And they call this the pigeonhole principle. Indeed, this comes back to Kimura. And that's very convenient for algorithmics, right? Most of you, or many of you, might be familiarized with the perfect phylogeny stuff, all this. So most models for doing evolutionary things in cancer assume this. But we have evidence, this and others, that we do have, homoplasy. We do have parallel events. We have reversal events. And in this estimate is in 21% of cancers you can find. This is just an estimate, this kind of events. The data is this, is real counts, sequencing, I assume that many of you are familiar with this, basically, and we focus on this. So our data is real counts. Right? There are colors that they will give us genotypes. And a very important distribution is the variant allele frequency distribution, which we call the site frequency spectrum in population genetics, the same thing here. And, this is used as a summary statistic for approximation computation, to estimate parameters and so on. And we expect that a clonal mutation, which is in all cells, heterozygotes, is above 0.5. And the shape of this is very informative, but there is a lot of noise, because we are talking about sequencing data, and we have noise. Using these BAF distributions, people have been trying to identify how many clones do I have. Remember, a clone is a set of identical cells with a common ancestor that share some mutations. So given the allele frequencies of these mutations, how many clones? So basically the idea is that you don't have recombination, remember. So mutations that share the same frequency is because they belong to the same clone. If you have 5 mutations, these 5 mutations can be in 1 clone or in 5, or in 3, or in 2. So that's the problem. And this is a very attractive problem, I would say, computationally, that has not been solved. There are 25 methods, but it still doesn't work well. Because, again, I think there is a problem of identifiability in the frequencies to distinguish different solutions. But in the cancer community, in the computational cancer community, people is using this. Of course, they assume there's a price, right, regarding the copy number, the infinite sites assumption, of course, right? And they are able to reconstruct something like a tree, with the difference here that the internal nodes are occupied, okay? It's not just stamped, right? Taxa, you have occupied nodes, internal nodes. Okay? But we have that in haplotype networks and things like that, so it's not new for evolutionary networks. The convolution, they often do this type of decomposition. When you have infinite sites, you can describe your tree with this matrix, and solving this can give you a configuration, and you can fit different trees to the same data, which is the observed frequencies of different mutations in different regions, and then you can solve it. And that's something that is used quite a bit, but in my opinion, is still a problem.
Because if you just look at the copy number and you don't take it for solid tumor, for example, taking into account the fact that the cell is coming from an ulcerated tissue and not from another tissue that is a little bit less ulcerated, and then they bring the same information in terms of copy number changes, so we cannot distinguish them, but can distinguish in terms of tissue.
But the clones are from one individual. You work one individual at a time. One individual, you have like a lung cancer. You're not gonna get a syringe, through a patient 5 times.
No, when you sample a tissue, you can have the sample.
Tissues are sometimes 5 millimeters. No, welcome to reality. No.
You cannot do that.
You get a colectomy, a piece of colon. Sometimes we get that. We do multi-regional. I'll show you some multi-regional. Right? No, but that's logistics is very complex, and often you get what you get. You have to, to beg pathologists, the doctors. They want to go home at 3. They don't want to spend their time carrying for you a piece of tissue. Reality is complex. So one thing is the theory. The other thing is trying to get the data. It's not that easy, but we can talk about this later. It's not normally you get all kind of samples. So you get what you get. You get a tube with something. Sometimes you don't even are able to extract DNA. It's frozen. Sometimes it's not frozen. Sometimes with OCT, different buffers. So, no, it's not that. It's not that thin. You don't get what you get, what you want to get often. But thank you. Okay, how do the cancer community goes about this? Well, they build these trees. I just told you, right? They get. Sorry, I didn't tell you. So, but to think, the cancer evolution community is dominated by genomics people. They are not evolutionary biologists. Well, I'm an evolutionary biologist, but so they propose things which are weird for evolutionary biology, like building a tree from different individuals for cancers, because different individuals are not connected. Different tumors from different individuals, they don't share a common ancestor. Maybe in the, I don't know, some mitochondrial leaf or something, but there is a lot of meiosis and recombination within. So this doesn't exist, this structure. They do these sample trees. Basically, they get bulk sequencing, allele frequencies, and then they get, from the allele frequencies, you can get, I don't know, Hamming distances or Cavalli's force stuff, like, from the frequencies, you get distances, you build a neighbor joining, and then you get a tree of the locations. So this is the same as building a tree of, New York, London, and Calcutta. What is that? Is there a phylogeny for London, New York, and Calcutta? No, of course there is not. Okay? There is a phylogeny for the ethnicities living in those cities, but locations don't have a phylogeny. Locations are not evolutionary units. But this is still, that's what I argue here. No? Okay, so bulk samples are not populations in general, unless there are some exceptions. So you have a monoclonal monophyletic metastasis, that's a single population, but in general, in evolutionary biology, when we identify for some feature population, some characteristics, so we have expectations for this beach to be different from that beach, the population to be a different clade, monophyletic, okay? But in general, we don't build trees for locations, right? But in cancer, this is done all the time. We did published some paper about this, some point, showing that if you do this, you get apparent homoplaces, wrong branching order, wrong branch lengths. If you mix things, mix clones from different locations and work with the locations instead of with the clones, it's still being published all the time in high-profile journals, this type of thing. So, to understand cancer evolution, we need to focus on the lineages. This is very obvious, but it's not done all the time. And lineages are the clones that we can infer from the sequence, or the cells, which are doing it. So when I got into this, I started to think, okay, well, we can get this clone phylogeny if we are able to estimate the clones, right? But underneath the clones, we have cells, and the cells are the individuals, and there is no recombination, remember. So the true history is the history of the cells. That's the history underlying. Clones are just clades, hopefully, okay, if they are estimated correctly. So I started to think about discontinuous, and think that we can do this. So I got into single-cell whole genome sequencing, and I had to open a wet lab 10 years ago. The thing is that when we get bulk sequencing, we sequence from millions of cells, and we get noisy data, not that noisy, but when we sequence the DNA from 6 picograms from one cell, we need to amplify, because we don't have enough material to build a library, a sequencing library. We need to amplify. And when we amplify, things happen. Okay? You get like a lot of heterogeneity in the coverage. Then you have allele dropouts, because basically you have 2 copies of the allele, right? The maternal and the paternal. If you miss it at the first replication, it won't be in your sequencing library. So you cannot sequence it because it's not in your library. Okay. And we have this, basically, individuals without heterozygous, you will only have one of the alleles in your sequencing library, then you put it in the machine. Of course, you can only get reads for one allele. Then you're going to think it's homozygote. So allele dropout. And then also, because there is some amplification here, you introduce errors. So you have false negatives and false positive errors. So it becomes very, very nice. The other thought that when we have cells is that, well, there is no recombination. So we can think of the cell phylogeny is basically the history of the maternal or paternal, because there is no recombination. They mirror each other, right? Because maternal and paternal, this is mitosis, not meiosis, remember. So you don't have any exchange of maternal and paternal material. So they are identical phylogenies. Okay? They travel together. So if you think of this, then you can think that we can go to our standard Markov models and build a diploid matrix, a Q matrix for instantaneous substitution rate among diploid genotypes, only in the case of no recombination. So that's what I thought, and we thought of this, thinking of unphased states where we don't know which of one of these is the maternal or the paternal. We can also work with a phase one when we care about, which one is the end. And then it's straightforward to fill this. And if we have this matrix, the Q matrix, we can calculate the likelihood of any tree, and then we can go to the likelihood. And for the sequencing error, so what I was thinking is that you can do something better than a very simple model which is just false positives and false negatives, because you can try to modelize the two sources of errors, the allele dropout and the amplification errors, that they are not exactly equivalent like this. Instead of thinking of mutation or non-mutation, we can think of the alleles exactly, whether you have an A, C, G, or T, and then you can build this, which is straightforward, observation models, and you can calculate genotype likelihoods and so on. And once I have this, then I thought, okay, I need to build a tree. Who's going to be better for this than Alexis? So I call Alexis and say, hey, Alexis, I have this model, this deployed genotype model and this error model. Can you do something with this? And say, of course. And he and Alexei implemented this in RACML. Okay. So voila, we have a likelihood, a program called Self-Fi to estimate single-cell sequencing trees, okay, where you incorporate the observational error, which is something that is ignored in species phylogenetics. You assume that your sequences are fine. Here you cannot, because there is a lot of. Other than that, everything is the same as in standard phylogenetics, I would say. This is very nice because then we have a probabilistic method. We have bootstraps, which are not used in cancer trees. They don't know what is a bootstrap. They just put a tree and they say, okay, this is my tree, and fine. So that was very nice. So using some simulation for this, simulating this observational error, we simulate sequencing reads because the depth of the sequencing is important, right? So we see that this method works well, and we're going to see here that we get very good estimates of these parameters because this Self-Fi estimates the allele dropout and the amplification error, these false positives, and quite well. And it is fast. Of course, that's what RACML is about, to do these things very fast. And well, we don't work with many taxa, many cells, because this is extremely expensive, right? To sequence, whole genome. It can be extremely expensive, so the amount of money is limited for doing this. So we applied to different data sets, and this paper was published at some point in a high-profile journal saying, well, you have two independent metastases, no way. And we applied this probabilistic method. We see that there is a single metastatic event. So therefore, this story of this late metastasis is not true. It's an early metastasis, and it's just one. So it changed the biological interpretation of the data. And of course, I trust much more a probabilistic method, some kind of a parsimony, infinite sites assumption problem. We applied also as a sanity check to a data set in which it's single cell data, but the way that they produce this data is to get a cell and make a clone. Expand this in the lab. This is hard work. You need, like a lot of people to do this, 100 single cell colonies, because often the single cells don't expand. But then you do bulk sequencing. And mutations which are in all the cells in the bulk sequencing have to be in the ancestral cell. So it's a way of getting single cell data without amplifying. Did I explain this more or less well? Okay. You expand your colony in the lab, and then you sequence the colony. Mutations in all the cells in the colony have to be in the ancestral cell. And then you work with that. So that data is very clean. There is no noise. And we apply, and we got this tree, which bootstrap support, very good. This is a development tree of hematopoietic stem cells. And we get I was very happy about this. We get an estimate of EDO of 0. Of course, it should be this way because there is no allele dropout in this data. Right? So it's doing what it has to do. Now, once we have a tool, I started to think. So a few years ago, I tried to be a marine biologist, and we went several times to Cape Verde and some place to get some conus, beautiful marine snails. And when I saw this figure in a seminal paper in the New England Journal of Medicine about multi-regional sequencing, I thought, well, that's what we do. So, of course, we can think that tumors are just evolution is just evolution. It doesn't matter whether it's within a body or among species. It's the same thing, right? So we can do this equation, and so the tumor can be and the metastasis an archipelago, and the continent the healthy tissue, We can describe the evolution, and we can do of cells in space using paleogeographical techniques. So we started to do this, and this is an old paper. I think it's a good story, so bear with me. Basically, we sequenced, this was an autopsy. So autopsies are the only cases where we are able to sample as much as we want. Okay? This is rare, right? But in this case, we got here 20 samples from 20 different regions. It's a colon cancer, so this is the primary tumor, lymphatic nodes, the liver, and lymphatic nodes of the liver, right? That was a few years ago. We used ion torrent, and we only got this number of SNVs for whole exome, very curated, very filtered. I'm not going to describe all the bioinformatics. There is a comprehensive bioinformatics pipelines to do this. You need to filter things, be sure, blah, blah, blah. That's not what I want to talk about today. And we did this study where basically, in this case, we used sulfide to get the topology, but we wanted to use also to estimate parameters. So in this case, let's try this, with this, okay? Without using the deploy model, just using a standard substitution model. So in this case, we had to assume a few things. We did some model selection, and we came up with a relaxed clock exponential growth as the best fit model for this data set. We estimate this, and so we were able to estimate, in this case, in this spacing, a single, this is not the case I showed before, it's a different one, right? A single metastatic event. So this is the metastasis more recent common ancestor. All the metastases have a single ancestor, so metastasis happen once, right? We map some, because we know this, right? These tools, substitutions, and the NDS, we identify some, this is the most interesting branch, is the branch leading to metastasis. Metastasis is the cause of more than 90% of deaths in cancer. Without metastasis, cancer doesn't heal. So we don't still have drivers of metastasis. We don't know where are the mutations, if these are mutations that cause, give a lineage the capability of colonize a new niche. And we identify very candidates. These genes are now candidates for being involved in the metastatic process because they are in the branch leading to this whole clade. We estimated, we have some prior for the mutation rate from some empirical in vitro estimates of colorectal. This is just a prior, right? We have some priors for this, for generation time. So having this, we are able to estimate the age, around 7 years, of the most recent common ancestor, right, of the sample, and the age of the metastasis, which, according to our calculation, this is the posterior distribution, happened around 2.5 years after the origin of the ancestral of the sample. Okay? In evolutionary biology, it's natural to talk about samples and ancestor of samples. In cancer, they say, this is the tumor phylogeny. No, this is the genealogy of your sample, which is not the same thing, of course. Okay? So this is our estimate, an early limit. In this case, we have an early metastasis. We also estimate diversification, skyline plots. We estimate these quantitative things, growth rates, doubling time, 200 days for this particular case. And also we wanted to understand some phylogeography. But the thing here is that I didn't say this, but we are working here with clones that we estimate from the frequencies, because this is bulk sequencing. So we estimate the frequency of the mutations, we estimate the clones with that, and clones can live in multiple locations. So therefore, we cannot use a standard discrete phylogeographic model, but we have models for ranges, right? So these models allow us to modelize the distribution of locations in which some species or some clone lives. We can estimate the ancestral ranges as well, okay? Type of models. And that's what we did. And we were able to come up with this story about this, which is the evolution of the spacing, and we even detect something which is called self-seeding, which sometimes seems like there's some attraction of the cells back to the primary tumor after the metastasis. But I think this was the first example in cancer where we have a very detailed study where we use standard for us, sophisticated for the cancer community methods to infer the evolution of the case. So that was, I think, a very nice study showing the community that we have the tools and we don't need to do many things different than we do in species evolution to have a more, sound approach to cancer evolution. We also try they have some solutions based on Sankoff, parsimony reconstruction, for phylogeography, but at the end of the day, they become very complex. A lot of people uses this, even last week there was a Nature paper, a big one using this tool, China, but it makes a lot of assumptions, and at the end, you have often too many solutions, so I think this is totally inferior to use a biogeography. Well, I'm not going to expand on this. If we use the sample tree, that's what normally they use, okay, just looking at the tree of the locations, we will infer at least 2 metastases. So answers change when you use the lineages and when you use the samples as units of your analysis. We also did some more things, more philo things. It's like, well, you know, there is some, I don't have time to explain this, but there is something called mutational signatures, which is based on the frequency of mutations when you look at the nucleotide 5' and 3', so you look at the 3 sites, and how often, depend on your context, we call this context-dependent models. And depending on this, you can detect different frequencies of mutations, and then you can assign the frequencies to mutational processes, like smoking, like deamination from the polymerase, like DNA repair. There are many of them, even chewing tobacco also, right? Chemotherapy, platinum, all kinds of things. And, well, they have the signatures 1 and 5 they call clock-like, because if you do a regression among individuals, among tumors from different individuals, then you see that the number of mutations accumulated in these signatures is proportional to the age of the individual. That's fine. So they said, these are clock-like. And I said, but you're missing something. You're doing a regression among individuals. That doesn't mean that within an individual, we have great heterogeneity. Okay? So they assume a clock all the time. So we developed this with Nico, with both Nikos, in which we apply the error model. It's a little bit more sophisticated because the error model is for the tips, but we also let it go a little bit further back, the error model, but it gets weaker and weaker, its influence. But we do a likelihood test for the clock, and we apply this to real data. And of course, here you have the p-values. We detected deviations from the clock in many cancer tissues and normal tissues. Okay? There is a lot of evolution in normal tissues. Now, there are a lot of studies on this. Remember, 500 million mutations per second. There is a lot of evolution going on in normal tissues. But here we saw that there is great heterogeneity, and when we estimate things, we cannot assume a clock by default. Same thing that we do in species evolution. Can we assume a clock by default? We can discuss about this, but I would say that we have the tool to test, to do model selection in this case, right, for the clock. Now what we're doing, and we're collaborating with Alexis Drummond and Kylie. Kylie is coming to my lab in October to the postdoc. We got in touch because they were interested in this model, the observational, the deep law substitution model and the error model, and they have developed a package called PhyLonco, okay, in BEAST, which is for somatic phylogenies, okay, somatic or cancer phylogenies, same thing. We did produce in our lab, you know, like probably the largest data set that is out there for single-cell sequencing data. Nuria is a postdoc in my lab, so we have these 400 cells. This is very expensive, bulk samples. This is a preprint now. And we got, you know, in this case, more pencils, but we focus on 7 of them. And this is whole exome, it's whole genome in this case, sequencing. At, you know, some depth, because this is a question, you have to, logistics and money are very important here in your experimental design. You sequence deeper or more in cells, you have to choose. But we think that this is kind of a compromise for us, sequences at this depth. And we were able to reanalyze the patient one, the one I told you the story about this biogeography, now with single cell data. Now the tips are not clones, are individual cells, and we are very happy that we again got more or less the same story, like a single origin of all the metastases, right? In this case, we have a benign tumor, and we can demonstrate that this benign tumor has nothing to do with the primary tumor in the colon. It's an independent event. This is obvious for any phylogeneticist, right? It's 2 independent events, right? High posterior probabilities for nodes. We map the mutations, blah, blah, and we estimate the time. This is a different data set, and our time estimate is very similar to the one I showed you before that we did with whole exome, clones, and this is whole genome cells. Okay? We get the same estimate. We're very happy about this for the same patient. And we applied to a bunch of patients, and we got some time estimates with the credible intervals around, you know, dozens of years, depends, 5, even 25, for the origin of the ancestor of the sample. In the demographics, we found some differences, but there was some instance that was repeated in some patients is that the tumor, this is one of the most clear cases, our estimate of the effective population size or diversity, if you want, simple, like was constant until 2 years before sampling, that exploded. And that pattern happened a few times, right? So that means that the tumor is more or less old and remains, indolent. The patient doesn't know the tumor is there, but doesn't grow for a long time, and suddenly, grows. That is important because that opens a window of opportunity for detection of tumors before they expand, in theory. Okay? We also estimate substitution rates, healthy versus tumor. This can be 1 order of magnitude higher, the substitution rate of the mutation rate of the tumor cells, which is not surprising. They divide faster, smaller generation time. They should accumulate mutations, and they do faster than normal cells. And we also got some estimates now of these technical parameters that we can get. As you can see, the ADO can be very important, right? Like up to 30% of the sites can experiment this allele dropout. So it's very important to use these models, statistical models that correct for the observational error. We do apply here some discrete phylogeographic model, which is a Markov chain, remember, for the changing among locations. That's one of the standard models, but in a Bayesian setting, so integrates over all the phylogenies, and basically we can estimate the location, posterior probabilities for the location of all the nodes, for all the patients, and we got, for patient 1, a very similar story that we got before with the clones, right? And for the other patients, it's more simpler because the sampling was much more limited, and we have 2 samples or 3, and there is not many opportunities for migration there. For this one patient, we got a similar story as we got before with a different data set. So it means that, to me, that these methods are reasonable and seem to work. At least they are consistent. And to finish, so now we move on, and for the first time in my career, I'm going to work with phenotypes. At the end of the day, phenotypes are the target of selection. Selection doesn't act on mutations. Selection acts on phenotypes. And in cancer, something that it is extremely important, this is recognized from a molecular biology point of view, from a biochemical point of view, is plasticity. Okay? Cells change their phenotype, function, without accumulating novel mutations. They do it all the time. So I want to study this phenomenon. I'm not the first one from an evolutionary point of view. There is a long tradition in the evolutionary community among phenotypic plasticity, right? Studying many taxa. And well, they are focusing on measuring this on what is the phenotype here. There are several levels, but one is gene expression. The most obvious phenotype you can get from a cell is which genes are expressing, because that's the function. So some authors have studied this, and they test phylogenetic signal, Pagel, right? Statistic, and so on, and they say, well, very few genes have a signal. This is one case where you have some signal. For this clade, have a different transcriptional pattern. There is a lot of noise. So transcriptional noise is over the place because there is no phylogenetic signal. They also say, well, if you have phylogenetic signal, the fact that related taxa are more similar phenotypically, what is phylogenetic signal? Everybody, I assume, right? And if you have signal, then the transcription state is heritable. This is not the quantitative genetic heritability. This is just how often your transcription, program is inherited by your daughter cells. And they say, well, not very often because, there is no much phylogenetic signal. That's wrong. Lack of phylogenetic signal is not lack of heritability. This is only true in a neutral model. Okay? In a neutral model, like a Brownian motion or quantitative traits, right? We can modelize this on a tree. This is well known. This has had decades of research. Evolutionary biologists have been using this to model things like size, quantitative traits. But we can, of course, do it for gene expression. So we can, that's a quantitative state. The only difference is that we have 20,000 traits, not one. And there, ideas for computational tricks to deal with 20,000 traits are welcome. So we are starting with this now. But indeed, here you have a phonetic signal. As you can see in this phenotype, it follows the tree. But of course, the action of selection erodes the phonetic signal. So those genes where you don't see phonetic signal are in fact the most interesting. They are not garbage, okay? And they are wrong. Luckily for me, I think, because this is very obvious from evolutionary point of view, but not from a genomics point of view. So we are not using OU models to estimate parameters like the strength of selection and whether you have one optima model selection or multiple optima for your transcriptional programs, right? Along the evolution of the cells. And we can do this because we have now the techniques to amplify, get the genome and the transcriptome from the same cell. So this is tricky, this is subtle, it fails sometimes, it's costly, money, but we're doing it in our lab. So to get our feet wet, we started with a cell line. This is human fibroblast. Well, we get data that we already have now, but not analyzed from tumors, but this is healthy cells and a cell line in our lab. We always have cell lines now to do experiments like this. So we got, this is just the 500 most variable expressed genes, and this is just a clustering of these. You can see that there are some structure in some of these genes, right? Some individual cells are closer than others. And of course, you can estimate a tree from the DNA data, and you use cell 5 for this, and then you have gene expression as traits. You normalize these gene expression values, blah, and we can apply models, right? In this case, I use R for doing this analysis. I do this very quickly with some R packages. So Lee and Rebel, Lou Hartman, Matt Pennell, all these, you are familiar with comparative phylogenetics, all these standard, assuming a single tree. Something we're changing now because we are incorporating this into phylong. We are incorporating these models into Bayesian somatics phylogenetics, but we can track and infer the ancestral expression of these genes, and we can estimate phylogenetic signal, lambda, pager lambda, for only a few genes show signal. So in cancer, they will throw away all these genes, and I think many of these are the most interesting, like this one. Right? We can fit multi-rate Brownian motions and see changes, right, in the Brownian motion. And we can fit the OU, or Steinulbach models, and estimate the strength of selection for some genes. This is one of the genes that doesn't have phylogenetic signal, but it has like a high value of strength of selection. And we can also use models like those implemented in surface to test for convergent evolution. Like in this case, these two lineages converge in the expression path. Right? So I think this opens totally a new view, new opportunities to understand selection and adaptation in cancer, ignoring DNA, just focusing on phenotypic plasticity. Because it's not my hypothesis, because it's been there around, is that we need to abandon the gene-centric point of view of evolution in cancer and pay more attention to the phenotypes. Because phenotypes, I'm going to repeat this again, is the target for selection, is not the genotypes. Selection doesn't act on genotypes at all, only acts on phenotypes. So this one map, one genotype, one phenotype might be wrong, and is wrong in cancer also. So many often cancer cells escape from drugs, just changing the transcriptome without changing their genotype. And we're going to use these tools to detect which genes are doing this, okay, in response to therapy. That's our brand. We're working on this. Well, I also have a beach where I live. Alexis, you've been there. Several of you have been there a few years ago. Well, this might not be surprising to you because I don't know if this is the first time I give a cancer talk to evolutionary biologists. Normally I do the opposite. I give an evolutionary biology talk to cancer people that they look at me like saying, what is this guy saying? It's like, well, I think maybe I convince you that we can use standard models for us, sophisticated for them. It's not very educated to talk about people when they are not in the room, right? But I'm not that well educated, so I can do it. They often like to hear things like uncertainty. And for us, it's natural, right? Because we want to measure uncertainty. But in some fields that things happen very fast and they are very competitive, uncertainty doesn't sound. But we can use applied standard evolutionary models. We can use fast tools like TELPI. We can use Bayesian packages like Philo now to estimate quantitative parameters, right? And do phylogeography and track the evolution of gene expression using tools that I'm not developing anything new here. I'm just getting the help of very clever people like Alexis and Alexei and people like this to implement this. And yes, we are producing the data. So we are completing the circle. We produce the data, we analyze the data, which is good for us. But the main take home is that statistical phylogetics, indeed, this might be obvious for you, but it's not obvious for the cancer community, can provide us a quantitative understanding of cancer evolution. And that's the end of my talk, I think. So, well, thank you to my team and to my collaborators, Moni, also. This is very expensive. I'm not going to. And I was going to put some sunglasses here. So this is Democritus, maybe the first scientist. So thank you very much.